Hello guys, this is OBED tutorials. Today we are going to talk about Krebs cycle or tricarboxylic acid cycle or citric acid cycle. So before that, let's do a quick recap on glycolysis. So in my previous video, discussed how glucose is being digested or metabolized to form pyruvate. So in the glycolysis, glucose is being broken down through some enzymatic reactions. In other way, catalyzed by various enzymes to form three carbon molecules that we call pyruvate, and then releases energy or ATP, and also release reducing equivalent, which is NADH. Okay, so after the pyruvate is formed, the Krebs cycle or the oxidation of the glucose occurs in mitochondria. Okay, but the pyruvate cannot enter the mitochondria, so it needs to be converted into molecule that we call acetyl-CoA. Okay, so for that we convert pyruvate to acetyl-CoA through some enzymes. We call them pyruvate dehydrogenase complex. They convert pyruvate to what? Acetyl-CoA. So this acetyl-CoA form is what we are going to talk about. It enters into Krebs cycle. So today we are going to discuss Krebs cycle. What are the reactions involved in Krebs cycle? Okay, so let's go. Let's start. And the question is, when do we call it Krebs cycle, or when do we call the cycle tricarboxylic acid cycle or citric acid cycle? So we call it Krebs cycle because it was named after the one who invented it or who elucidated the sequence of the reactions in 1937. So the man is called Sir Hans Adolf Krebs. So the cycle or the process was named after him. Okay, he's a German to British biochemist. Okay, and also we call it citric acid cycle because the first molecule produced in the cycle is the citrate or citric acid. It's formed as a result of the acetyl-CoA reacting or combines with oxaloacetate to produce the citrate. So the citrate is the first molecule produced in the TCA cycle. So the term citric acid cycle is as a result of that first molecule produced in the cycle. And also we have tricarboxylic acid cycle. We call it tricarboxylic acid cycle because the chemical structure of the citric acid contains three carboxyl groups. As you can see, it contains three carboxyl groups. That's why we call it tricarboxylic acid cycle. So because of the three carboxyl group the citric acid contains, that's why we call it tricarboxylic acid cycle. Okay, so now let's enter the process of the Krebs cycle proper. So the Krebs cycle, the process occurs in mitochondria. Okay, it occurs in mitochondria. It makes use of oxygen. So now what happens is the acetyl-CoA from the conversion of pyruvate to acetyl-CoA. The acetyl-CoA produce urea to this oxaloacetate, which is 4 carbon compound, and then acetyl-CoA is 2 carbon compound together with water to form 6 carbon compound that we call citrate or citric acid. In so doing, the CoA molecules on the acetyl-CoA will be eliminated, will be removed. Okay, so this reaction is catalyzed by an enzyme that we call citrate synthase. Citrate synthase. The moment we hear synthase, it means that it doesn't involve the use of ATP. Whilst synthesis, it involves the use of ATP. So the moment we hear synthesize, so citrate synthase, we are synthesizing what? Citrates without the use of ATP. Okay. So this citrate will undergo isomerization. It will undergo isomerization, okay, to form a compound that we call isocitrate. So isocitrate is the isomer of citrate. That's why we call it isocitrate, the isomer of citrate. And this is catalyzed by an enzyme called aconitase. Okay, aconitase. And then the isocitrate is also reduced to, mind you, the citrate is 6 carbon compound, and then it's reduced to 5 carbon compound. So we call it, the compound form, we call it alpha ketoglutarate. This is the alpha ketoglutarate. So this reaction is also catalyzed by an enzyme called isocitrate dehydrogenase. Isocitrate dehydrogenase. So what happens here is that the NAD plus, which is oxidating form of this NADH, will reduce the isocitrate to alpha ketoglutarate. So it will just remove its a little bit of decarboxylation. It's a little bit of decarboxylation. This one, the outside group, is being removed from the isocitrate to form carbon dioxide here. And then the NAD plus will be reduced to NADH. So all this happened by the action of enzyme we called isocitrate dehydrogenase. So now we formed our 5 carbon compound, which we called alpha ketoglutarate. And this alpha ketoglutarate will also undergo a reduction reaction. So here what happened is OA molecule is being added to the compound, and then carbon dioxide is removed from alpha ketoglutarate. So we form 4 carbon compound here. That what we called succinyl-CoA. So we added CoA molecule to the alpha ketoglutarate and then remove carbon dioxide from the same alpha ketoglutarate. So we form succinyl-CoA, which is 4 carbon compound. And this process is being catalyzed by an enzyme called alpha ketoglutarate dehydrogenase complex. Okay. Alpha ketoglutarate dehydrogenase complex. So now here, from succinyl-CoA to succinate, we are going to synthesize what? Succinate. You get it? We are going to synthesize succinate. And here it involves the GDP to ATP, GTP. So it's more of using of energy. You get it? It's a more of use of energy. So now what happens here is succinyl-CoA will be converted to succinate. They are all of 4 carbon compounds, but this process is catalyzed by an enzyme called succinyl-CoA synthetase. Succinyl-CoA synthetase. It uses GDP. GDP is also an energy. It's an energy producing compound. Normally, it produces 1 ATP of energy. It produces 1 ATP of energy. That's why we have it. Synthetase. If it were to happen without the use of energy, it would have been synthase. But now it uses energy. It's synthetase. Okay. So now we form our succinate. Succinate will also undergo reaction that leads to the formation of fumarates. So now succinate will be reduced to fumarate by the action of FAD, FAD plus or FAD2, but FADH. So FAD will be reduced to FADH2. So this process is also catalyzed by an enzyme called succinate dehydrogenase, and then the fumarate too will be converted to what? The malate. What is the malate? They are all 4 carbon compounds. So from succinyl-CoA to oxaloacetate, they are all 4 carbon compounds. So fumarate will undergo dehydration. No, fumarate will undergo hydration. We add water to fumarate to form our malate. So this process is also catalyzed by an enzyme called fumarase. So we add water molecule to fumarate to form malate. Then malate also undergo reaction to form oxaloacetate. Okay, which is also catalyzed by an enzyme called malate dehydrogenase. So the oxaloacetate form here will also combine with the next acetyl-CoA to undergo the same process again. That's why we purposely call it cycle. It goes round non-stop. It goes round. Okay, it goes round. So now let's look at some things here. We have NAD plus to NADH 1, NAD plus to NADH 2, and then we have 1 GTP, and then 1 FADH2, and then 1 NAD plus here. So we have NADH 3 of them, and we also have 1 GTP and 1 FADH. So these molecules, most especially NADH and then FADH, will enter into a process that we call electron transport chain. So this, we call them reducing equivalent. This molecule will be converted to form ATP in a process that we call electron transport chain. Okay, so now my previous video was on electron transport chain. How does this reducing equivalent, which is NADH and then FADH, will be converted into ATP? Okay, it's the same process here. Then we talk about glucose metabolism to produce acetyl-CoA. Acetyl-CoA will added to oxaloacetate to form citrate. Citrate to undergo isomerization, form isocitrate, and isocitrate will also undergo reduction by removing one molecule of carbon dioxide to form alpha ketoglutarate. And alpha ketoglutarate form here will also be reduced to succinyl-CoA. From succinyl-CoA to succinate, also from succinate to fumarate, and fumarate to malate. Then we get to oxaloacetate. So the process will continue again. So mind you, we call in biochemistry, we call this process, this molecule here, in biochemistry, we call these molecules, we call them metabolites. Why do we call them metabolites? We call them metabolite because this molecule is not is an end product of certain reactions, but it can also be a starting material for other reactions. Okay, so please, this is OBED tutorials. Kindly subscribe, share, and like. Please make sure you turn on the notification button so anytime I post a new video, you will get it fast and appreciate it. Thank you very much. Bye bye.